This soothing lullaby gently lulls the child to sweet slumber every night, even as the sleep fairy tiptoes into the room, and oh so softly, weaving a spell of magical, sweet dreams. But not so tonight. Tonight, something nasty has raised its ugly head and disrupted the sweet harmony. Fever is a frequent occurrence in children. It erupts through a rise in the body temperature, often accompanied by cold, cough, headache, body ache, poor appetite, and difficulty in sleeping. Fever can be a traumatic experience for the innocent child, as also for the parents, who spend many sleepless nights trying to comfort their precious one. Their immense concern often clouded with misconceptions, culminating in what psychologists term as the fever phobia. Fever management in children is definitely one of the pressing challenges in parenting, and most anxious parents seek immediate medical attention for their children, seeking you, the doctor, as their trusted partner to help meet this challenge. Fever occurs as an acute phase response to infection caused due to the invasion of exogenous pyrogens such as bacteria or viruses, affecting either the whole body or a specific body part and resulting in mild, low-grade to high-grade fever.
The hypothalamus is located below the thalamus and constitutes just 1% of the brain. It is very important in controlling many vital functions of the body, including the regulation of body temperature. Because it regulates the body temperature, the hypothalamus is supposed to be called as the body's thermostat and is very, very important in the genesis of fever.
However, the pertinent question is, if the fever has to be tackled centrally, is there a drug that can reach the right place and exert the right action? Sensing the gaping absence of such a drug in a market crowded with NSAIDs like paracetamols and ibuprofen, the team of medical specialists at Blue Cross Laboratories, one of India's leading pharmaceutical companies, struck a marvel in discovering a reliable breakthrough for fever management and introduced Meftal P. Meftal P, made with active key ingredient mefenamic acid, is unique for its double blockade antiprostaglandin action inside the hypothalamus, thus reducing temperature and controlling the fever centrally. To understand the workings of mefenamic acid as a high-quality antipyretic, let us delve a tad deeper and trace the path that makes fever happen.
Chemicals called lipopolysaccharides and peptidoglycans are present in the cell walls of the invading pathogens. When these pathogens enter the body, these chemicals stimulate a nearby macrophage by interacting with its CD14 receptor. The now activated macrophages liberate interleukin-1 and tumor necrosis factor alpha, which are briefly known as IL-1 and TNF-alpha. These chemicals are endogenous pyrogens which go into the general circulation and enter into the circumventricular parts of the brain as well as the spinal cord, since these are the areas where there is the least resistance offered by the blood-brain barrier.
Interleukin-1 and tumor necrosis factor alpha enter the hypothalamus and activate the microglial cells to generate prostaglandins, particularly PGE. The cell membrane of the microglial cell has two layers of phospholipids placed this way. The head, with its phosphorus component, is on the outside, and the two tails containing fatty acids face each other in the center. The fatty acids present in abundance in the tails is arachidonic acid, derived from all the lipids consumed in vegetarian and non-vegetarian foodstuffs, excluding marine sources. The enzyme phospholipase A2, released during fever, breaks down the phospholipids by cutting the neck and separating the head from the two tails. The arachidonic acid released interacts with the enzyme cyclooxygenase, popularly referred to as COX, produced by activated microglial cells. The COX enzyme is a channel into which the liberated arachidonic acid enters. On interacting with an amino acid, tyrosine at 385 position, present in the COX channel. The arachidonic acid gets converted to the notorious fever-causing PGE2. Thus, the concentration of the PGE2 increases in the hypothalamus, turning up the hypothalamic thermostat, and the body temperature rises.
The key to understanding the mechanism of fever lies in unraveling the COX puzzle in the brain. Now in the brain, we have three variants of COX. We have COX-1, COX-2 and COX-3. COX-1 is present in small quantities in every part of the brain. COX-2 is also present in small quantities but primarily in the cerebral cortex. In a normal person, the COX-2 enzyme is important for transfer of information within the brain and for the memory functioning. COX-3 is the third enzyme which is also present only in the cerebral cortex. But there is an issue. The COX-3 enzyme quantities are just 5% of COX-1, which as it is less present. Secondly, the COX-3 enzyme ability to make prostaglandin E2, that is PGE2, which is responsible for fever, is a mere 20% as compared to COX-2. More importantly, the COX-3 enzyme is not present in the hypothalamus. Since the hypothalamus is the thermostat of the body, the COX-2 enzyme is more important for the role in genesis of fever.
So, the trump is in quelling COX-2. And mefenamic acid, with its powerful double blockade antiprostaglandin action, is the only antipyretic that quells COX-2 in the hypothalamus. Action 1. Mefenamic acid inhibits the COX-2 enzyme centrally, that is, in the hypothalamus, thus inhibiting prostaglandin synthesis. Action 2. Mefenamic acid is the one and only antipyretic that blocks the EP receptor sites and inhibits the already released prostaglandins, fostering quick relief. Action on the EP receptors is essential, as only after the PGE sits inside the EP receptors, the body temperature is raised, so much so that a landmark experiment showed astounding results such as, if these receptors are removed from the hypothalamus, fever state will actually be non-existent.
To reduce the high temperature in fever, basically the thermostat needs to be reset. For this, the antipyretic has to penetrate into the brain. Today, there are only two such agents which can penetrate the brain. One is the traditional paracetamol and secondly it is mefenamic acid. Studies have revealed that paracetamol has weak action on COX-2, which is responsible for the fever, although it has strong action on COX-3. But mefenamic acid has weak action on COX-3 but very strong action on the villainous COX-2.
Also, being the only antipyretic to enter the hypothalamus, Meftal-P scores a master stroke. It exhibits superior control of fever compared to paracetamol. It has a long-lasting effect for 8 hours, much longer than the others, like approximately 4 hours in the case of paracetamol and 6 hours for ibuprofen. Little wonder then that mefenamic acid is hailed by the Indian Academy of Pediatrics textbook of pediatrics as an effective option for fever management, more effective than ibuprofen. Even Martindale and Therapeutic Drugs textbooks recommend mefenamic acid for fever management. Every 5 ml of Meftal-P is power-packed with mefenamic acid 100 mg, tackling fever proactively, deftly, at the center, and all this with a delicious mango flavor. So now, it's time the relieved parents got back to singing that sweet lullaby, cradling and rocking the child in their sweet, engaging warmth, letting the sleep fairy gently guide their precious one to a relaxing night's sleep. For Meftal-P comes with the solid assurance of sustained nighttime control of fever for 8 hours. Meftal-P, a dual-action antipyretic with the highest therapeutic efficacy, empowering you with the most potent formulation for fever management. Meftal P, the first choice antipyretic from the House of Blue Cross.